that's on the eye tomorrow. All right, so for lab tomorrow is a dissection day. So remember the closed-toed shoes. It's getting to winter time now. Not too many people are wearing flip-flops anyway. Closed-toed shoes and gloves. And we're also going to do eye and ear function. So we'll be testing hearing with tuning forks and looking at the eye test charts, which you can do today if you want to, but most people were looking at the model and stuff for the eye quiz. And looking at negative images, look at something red and then look away. You see the same shape but in a different color. Yeah. All right, so we are on in your eye, or not in your eye, but in front of your eye. We are on page 114 of your lecture notes. So this is where we left on Tuesday with the diagram of the wall structure of the eye. And we just kind of identified the outer pigmented layer and the retina. Okay. So looking at the cells of the retina, this screen here is kind of as if you were looking over here, as far as where it's taken from, what it's taken from. So I'm going to kind of redraw it on the board and then center here in the pathway of the eye. So the outer layer of the nervous tuning was formed of pigmented epithelia. These are cuboidal cells. The base of the cuboidal cell would be up against the choroid, where the choroid is the layer of the vascular layer that's very rich in blood vessels and capillary. So this would be the choroid layer, what I've drawn here in red. And then we would have the outer pigmented epithelia have microvilli facing towards the pathway of the light. And remember the major purpose for having microvilli?
Increase surface area.
Increase surface area, all right? These outer pigmented epithelia, sometimes abbreviated E, have two functions. The only function I gave you on Tuesday was that they absorb the light and they prevent it from being reflected back into the eyeball and bouncing around on the inside of the eye. And so they have melanin, that's their pigmented component. They're going to have melanin here in order to absorb the light. These cells, you can have melanoma. These cells can develop a cancer. Just like the skin melanoma, you can have melanoma in your eye from these cells with too much exposure to ultraviolet light. Nestled into these cells, as you can see over here and in the diagram that you have on your lecture notes, we have the tip of the photoreceptor. And I'm going to draw in a rod. So instead of a kind of cone shape, it has a shape like this. And those have discs. We're going to talk about the discs in about half an hour. And nucleus. And then their axon terminal components. So this is the receptor. This is not the first order neuron in our pathway. Okay? This would be the receptor. So this is a rod. The cones have more of a cone shape. They would be the purple cells that you see in this diagram. Obviously they're not purple in your lecture notes unless you printed them off from online. So these cells are going to synapse with the first order neurons. And these are bipolar cells. Remember we had bipolar cells in the olfactory pathway, and we have bipolar cells in our retina, and these would be the first order neurons of our visual pathway. And then those bipolar cells, the second order neurons, are identified as ganglion cells. And their axons are actually what form the optic nerve. And those are what end up forming the optic nerve, the optic tract, and end up in the thalamus. Now I'm going to reverse that because in actuality the pigmented epithelia are on the outer part. So I'm going to redraw this where it would sit in our light pathway. So remember in our layering we had the sclera, the choroid, the pigmented epithelium, and the nerve cells. Okay, and so in our direct line of pathway, light pathway, notice I've drawn an indented area right here, and that's identified as the fovea centralis. So you can see it in the diagram on the bottom of page 108. It's just labeled fovea there. But it is the fovea centralis. Remember we had the fovea capitis on the femur? And it was that depression in the head. So this indented area is where we have the highest concentration of cones. Those are the purple cells that you saw up here. And cones, as we'll see in a few minutes, have pigments that are sensitive to various wavelengths. So we see color. In this area, we only have cones. There are no rods. So where we have our best focus, you'll notice when you look at me, I'm in focus, I hope. But you can also see the screen, and you can see the door over there, but while you're looking at me, those objects are not in focus. You look at those objects, and I go out of focus. So what happens is when you look at an object that is in focus, the image, light waves, are being focused at that fovea centralis. Okay. Light is also landing on other areas of the retina, so I can see them, but they're not in focus. And as we move laterally, we decrease the number of cones and increase the number of rods. All right, so this has the highest area of cones. It actually has no rods at all. So I'm going to reverse this image and put in the cone, all right? Put in the cone structure. And so we're going to look at this view here. So we're going to have our cone. I'll make it a little bit smaller. That will be a cone. Actually, I'm sorry, let me do it in the same color so you guys don't get mixed up. And then we'll have our bipolar cell. Then we'll have the ganglion. And I'm doing this so that you recognize that the ganglion process is the structure that is closest to the light wave. So as these ganglion nerve fibers pass out of the eyeball, they form the optic nerve. So the very surface is where we're going to find our vitreous humor, the jelly, grape jelly-like material that's in the back of the eyeball. That's in contact with the light. So the light has to pass through all those nerve fibers, pass by the bipolar cells, pass by most of the photoreceptor cell to actually get to the light-sensitive pigments within the cone or the rod. So let me turn this light off briefly. So if you look at this diagram, again, this is just an enlarged version of the one that you have on your lecture notes. So the light is coming in this direction. It has to pass through all of these nerve fibers fibers of the ganglion cells. And then it has to pass in between the bipolar cells. There are some additional cells that are oriented in a horizontal position known as horizontal or amacrine cells. It has to pass between their fibers. It has to get down to this area of the rods and the cones. Now, if you look at the eye of an animal that lives deeper in ocean water where there's very little light, okay, like a squid, that's actually the reverse. The receptors are out on the surface next to the vitreous body, and the ganglion cells are farthest away from the light. So maybe if we didn't have this pattern, we'd have way too much light in our eyes. They get bleached out with all that light. Okay, so I told you one purpose of the pigmented epithelia was to absorb that extra light. Another purpose is as we bleach out the photopigments, the pigmented epithelium functions to endocytose those. They'll take up these discs and break them down and recycle the pigments back to the rods and the cones. So they're sitting here just kind of gobbling in this material. They don't really use it for nourishment. And then recycling it and sending it back to the rods and cones. So that's both of those, their functions. Okay? So if this is our best focus point and only cones, that means, have you ever noticed at night, you look at something that's kind of just a very faint light, you can't see it, like a star, and then when you look away, it shows up again, but it's not in focus, so you look at it to try to bring it back into focus and it disappears. I have a light over our door that's from the smoke detector in my bedroom. And when I look at it at night, it goes away. And if I look slightly 4 feet or so to either side, I can see it again, but it's not in focus because it's not landing on my fovea. But when I look at it again, it disappears. And the reason for that is the rods are sensitive to low intensity levels of light, but not to color. The cones require a higher intensity of light, and so they're not activated by dim light. When there was a comet in this area about 15 years or so ago, and we took the kids out to the countryside where there wasn't so much ambient light, about 2 o'clock in the morning, and I told them, don't look directly at the tail of the comet, look kind of away, and then there would be enough of the rod that light was landing on to be activated. So try that sometime at night, okay? Find a dim star and look at it, and you'll see it better if you look off to the side. And that's because the energy will be landing over here or over here where we have more rods. So as we move up to this area, we have decreasing cones and increasing rods until up here we have only rods. All right? Now, let's focus at this area, no pun intended. Notice that we have only the nerve fibers. There are no receptors there. And this region is known as the optic disc. It's also known as the blind spot. Because when light lands on that, when the focus is on the optic disc, there are no photoreceptors to be activated. So an image is not created. If you look at the bottom of page 114, you'll notice an X and an O. So here's the optic disc. All the ganglion fibers are coming together to form the optic nerve, but no photoreceptors. All right? So there's an X and an O. I can only blink my left eye. I told you if I do my right eye, I have to hold it down with my finger. So if you're looking at this with your right eye, you want to focus on the X. Okay? If your left eye is open and your right eye is closed, you'll want to look at the O. Otherwise, it won't work. So close the whichever eye. I'm closing my left eye. And on page 114, I'm looking at the X. I can see the X. As I bring it closer, the O disappears. So I can see the O here. I'm not looking at the O. I'm looking at the X, but in my peripheral vision, I can see the O. All right? So my focus is